Hi, everyone. I am really excited to have Leonora speaking with us today. She's done some really cool work and has some really intriguing ideas about the assembly and function of microbial communities associated with plants. Leonora got her undergrad degree from UC Berkeley and her PhD from the Department of Organismic and Evolutionary Biology at Harvard. She then was a James S. McDonnell postdoctoral fellow in complex systems at MIT before starting her lab at Boise State just recently in January. Thank you, Leonora, for taking the time to speak with us.
Thank you so much, Karen. Thanks for the introduction. I'm really happy to be here and to talk with all of you about some of my work. I'll be talking about these unusual plants, these carnivorous pitcher plants, and the microcosms and little communities that are inside of them. So in an ecosystem like a rainforest, there are thousands of different species that are all interacting with each other. And there's predator hunting their prey. There are trees that are primary producers and are competing for sunlight, for water, for nutrients. And then there's decomposers that are breaking down the plant material and recycling those nutrients. And among all of these many species, the vast majority are microbial. We can't even see them without a microscope. But despite their very small sizes, they can drive the biogeochemical cycles of our planet. So they can have really large effects on ecosystems. And then if you zoom in on, even the tiniest leaf on one of these big rainforest trees, you would see that it has this associated community of bacteria and fungi that are living on and inside of it. And these microbes can actually affect the fitness of this large host. Even within our own bodies, on and inside of our own bodies, are microbial ecosystems. For example, our gut microbiome, which can really affect our health and even our state of mind. So we're starting to understand that these microbial communities have really large effects in our world, despite their very small sizes, but they can be difficult to study. And for example, a rainforest can be hard to study all the different microbes living there because it's just so vast and there's so many different sort of types of subhabitats. And then something like a gut is pretty difficult to manipulate. We can study it, but it can be hard and not very experimentally tractable. So I think we need to find more model ecosystems that we can really study and manipulate and probe to understand more about how these microbes, how these communities form, how these microbes act within these different ecosystems. I'm really curious about this question of what shapes microbial communities. And I'm going to walk you through some of the ideas that we know from community ecology, from theory. We know that we start with a source pool or a regional pool that's influenced by geography. So different microbes can exist in different parts of the world. Then a subset of these will be able to disperse into a new habitat. And then only a subset of those are going to be able to colonize and thrive in a new environment. And this is environmental filtering, where characteristics of that particular habitat restrict what is able to survive and thrive. So this could be things like the acidity of that environment, the nutrients available, things like that. Then over time, we can have successional processes that lead to different community compositions. So we can see maybe primary colonizers giving way to better competitors over time. And we also know that historical contingencies can affect communities. So these are things like a random weather event or a nutrient pulse, or even which microbe happens to arrive first. So these are sort of random and stochastic things that are difficult to predict, but these can have effects on how communities form and what the composition is of that community. Then we know that these different community compositions can have differences in ecosystem functions. And ecosystem functions are things like the productivity of that system, or the way that nutrients move through it, like carbon, nitrogen. So I like to think about all these different processes that may be influencing and shaping microbial communities, but there's a lot that we still don't know about in which context these are acting and what might be more important for microbial communities. So I wanted to develop a system where it would be easier to ask these questions and to understand more. And I think that carnivorous pitcher plants are a really great system for studying this. So I'll introduce you to them. This is a modified leaf, and it's been modified through evolutionary time into this cup shape. And this cup holds water. There's a little aquatic system inside of it. And that water is a combination of rainwater and plant secretions. And the plant produces nectar around the lip and under the top of this lid. And that nectar attracts prey, mainly insects. And they come to feed on that nectar. And then the insides are very slippery and they fall in and they drown in the pool inside. But even though these plants have evolved to attract and trap and eat insects, they also host a whole little ecosystem inside of them. So these aquatic pools have insects that you only find associated with pitcher plants. They're specialized for this type of habitat. And it has protozoa and rotifers and fungal yeasts and bacteria. And there's predators attacking prey. There's primary producers, there's decomposers. All of these different processes are happening, but on this very small scale. And when pitcher plants open, so they're like a passive pitfall trap, and they're sterile, or at least close to sterile, until they open. And then once they open, a community colonizes from the surrounding environment. And it's thought that that community, that microbiome that forms in the pitcher fluid, helps the plant to break down and digest its prey and access nutrients from its food. And so in this way, the pitcher plant is also sort of an analog of a gut, right? It's a plant's version of a gut. And I think it's a nice system that you can access and manipulate and think about as sort of this host-associated microbiome. So there's a variety of reasons why I think these are great little model ecosystems. One is that it's a small, easily defined area. And when you're studying community ecology, sometimes it can be really difficult to determine like what exactly is your community of interest. So in a pitcher plant, that's pretty easy because it's just this little aquatic pool. And it's almost like this aquatic island in a terrestrial sea. So you have like these little islands that you can study. It has a tube-like shape, so that's nice if you want to bring them into the lab. They're already sort of like a tube, so it's not that different as, say, it would be if you were trying to study the ocean or a different type of system. They're naturally replicated, meaning there's a lot of them in the same habitat. So you can have many different pitchers existing together. You can have many different pitchers growing on the same plant. And they have this nice host microbiome association. So we think that those microbes living in the pitcher are really helping to break down the prey and access nutrients. And because of this, there's a pretty clear ecosystem function. So it can be difficult to sometimes determine what are the relevant functions in a system or what you might be interested in. But because of this host microbiome association and the fact that the pitcher plant is sort of formed to attract, trap, and digest prey, it has this nice way of linking the microbiome to something that's relevant for the host, which is the breakdown and degradation of this insect prey. So here I'm showing you an evolutionary tree of flowering plants and all the different times that carnivory has evolved. So you can see that carnivory in plants has evolved multiple different times. And I'm going to highlight the pitcher plants. So here is the Nepenthaceae family. This is in the core eudicots, and these are found in Southeast Asia. This is the Cephalotaceae family, and there's actually only one species, and it's found in Western Australia. And then this is the Sarraceniaceae family, and there's 3 different genera, and these are found in the Americas. So you can see that all 3 of these are in different orders. And these are really beautiful examples of convergent evolution, where the same structure and function has evolved totally independently in really distantly related groups. And just to highlight how distant these are to each other, the ones I'll be focusing on today are Nepenthes and Sarracenia, and they're as distantly related as cacti are to blueberries. So you can see that there really isn't a common ancestor that has these characteristics. It's totally independent evolution. Today I'm going to talk about two main projects. One is a global characterization and comparison of the Nepenthes and Sarracenia pitcher plant communities to understand more about who's there. And the second is a laboratory experiment to examine environmental filtering, historical contingency, and changes in ecosystem function. Before I begin, I want to acknowledge that I was sampling from and working in the ancestral lands of many different Native people. The Kadazan Dusun in Sabah, Borneo, the Malay in Singapore, the Wabanaki and Nipmuc in Massachusetts, the Chatayakni, Muskogee, Timucua, and Seminole along the US Gulf Coast, and I currently live in Boise, Idaho on Shoshone-Bannock Native land. So what organisms live in pitchers? Before I started this work, it was pretty well known the different insects that colonize pitcher plants, but not a lot was known about the microbes. So I wanted to be able to understand that more. We collected over 500 different samples in North America, Southeast Asia, and Australia from pitchers, and then also from their direct surroundings. So from the bog water or from the soil that was directly around the plants. And then I did metabarcoding of the bacteria and the eukaryotes living in these pitchers. So this is using 16S and 18S ribosomal RNA genes to be able to understand who's there in these different samples. And then from a subset of our samples, we did shotgun metagenomes. And this is to look at the functional potential of the whole community. So it's basically breaking it all up and looking at a subset of all the different genes that are in there to understand more about what functions it may have. This is me sampling in Borneo. I was really fortunate to work with some great Malaysian graduate students. And you can see I'm wearing leech socks. This forest has these ground leeches that will climb up your legs. So you have to be really careful and wear leech socks so you don't end up losing a lot of blood. And then this is me sampling along the Gulf Coast in the US. So this was in Alabama. And these are these beautiful longleaf pine savannas, which are actually a pretty threatened ecosystem now. A lot of them are being drained and developed. And so there's not a lot of habitat left for these cool plants. What did we find from this sort of global characterization, looking at what's there? Really broadly, what we found was that in terms of eukaryotes, there's mostly mosquitoes, midges, mites, fungi, protozoa, and algae that are living in these pitchers. And in terms of bacteria, there's mostly proteobacteria, Bacteroidetes, actinobacteria, and acidobacteria. And one of the questions I was interested in looking broadly across these different systems was thinking about if succession leads to change in pitcher communities over time. So understanding more about the successional process within pitchers. To look at this, we focused on just one species, Nepenthes mirabilis, and I worked with an undergrad, Elizabeth Benson, to do this study over time. So what she did was she found a lot of closed pitchers before they had opened, and she followed them over 4 different subsequent weeks. So a week after they'd opened up to 4 weeks after they'd opened. And I'm going to be talking a lot today about either OTUs or ASVs, and that stands for Operational Taxonomic Unit or Amplicon Sequence Variant. And if you're not familiar with these terms, basically you can think of them as species or as phylotypes. So what I'm looking at here on the y-axis is basically like the species richness. And then here, this is the different weeks. And what we see is that for the bacteria, there was a strong increase over time. And then there was also a small increase for the eukaryotes. In terms of beta diversity, so this is an NMDS plot. And if you're not familiar with these plots, what's important to know is that each dot represents the whole bacterial community found in one sample, in one pitcher. And if those two are close to each other, it means that those communities are similar. If those dots are far away from each other, it means that those communities are less similar to each other. And here I've colored the different points by the weeks when they were sampled. And then I've connected them by the median point in the middle. So you can see that it goes from week 1 to 2 to 3 to 4. So there is this progression happening over time in terms of how similar the communities are to each other. Those in the first week tend to be more similar, and they're less similar to the ones in week 4. We see this sort of progression through weeks. So it does seem like there is a successional change that's happening in this community composition across different weeks over time. So here I'm going to bring you back to this slide that I talked about earlier, and we can see that this successional process is important and is happening within these systems. So does environmental filtering alter pitcher communities? I was sampling from 8 different Nepenthes species and 6 different Sarracenia species. And you can see that these different species have sort of different shapes, different forms. There's a lot of different differences among the hosts. And what we found was that within each genus, within the Nepenthes and within the Sarracenia, host species identity did influence the composition of the pitcher communities. So this species did, have significantly different community from, say, this one. And we were wondering what might be driving this. I'm going to give you a couple examples of characteristics of the pitchers that we think are driving these composition differences. In the Nepenthes, the pH, or the acidity of the pitcher fluid, is one of the biggest drivers. And that's because Nepenthes can get extremely acidic. They can get all the way down to a pH of 1.5, super, super acidic. But these communities still seem to be pretty happy, and I would even find mosquitoes swimming around in these very acidic pitchers. And it's known that the Nepenthes plants can actively acidify their pitchers by excreting hydrogen ions into the fluid. So what we found, here's another NMDS plot, and now these different points, these different bacterial communities, are colored according to the pH or the acidity of the fluid. And you can also see that it's sort of mapped on here, so it follows this trend. And what we see is that there is also this correlation between acidity of the pitcher fluid and the similarity of the communities. And this is it's highly significant with a Mantel test. I want to emphasize that this effect is much stronger than the effects of dispersal. So for example, 2 pitchers that are next to each other in Borneo, if one was very acidic and one was less acidic, those communities were less similar to each other than the very acidic one would be to a very acidic one that was on Singapore, on the island of Singapore. So it seems like this type of environmental filtering is having a pretty large effect. In the Sarracenia, the ones that are found in North America, the acidity was not such a strong driver, and that's partly because they don't get down to these really low acidities. Instead, we saw that differences in the shape seem to be driving differences in the communities, and shape is correlated with volume. And so we have some pitchers that are these narrow, tall, fluted style pitchers, and others that are more squat and round. And what I found was that in general, the tall, fluted pitchers, those communities would be more similar to each other than the ones that were found in the more squat and round pitchers. And this also is much stronger than the effects of dispersal. So for example, Sarracenia purpurea and rosea, these two, the blue and the green, one is found in Florida. I was sampling it in Florida, and the other one I was sampling in Massachusetts. But these two are more similar to each other than the one from Florida is to other plants that are growing maybe in the exact same field in Florida, the same pine savanna. So this also seemed to be a strong driver. So what we can see from that is that environmental filtering definitely is playing a role in this system as well. And it's playing a pretty strong role, stronger than the effects of dispersal. So it's definitely having an effect on what the community composition looks like. Stepping way back, instead of comparing different species to each other within the same genus, I wanted to look really broadly at what type of organisms tend to colonize pitcher plants in general. And there's kind of a nice natural experiment, the fact that these two groups are convergently evolved. We could ask, is the convergent evolution of the hosts driving convergence in the associated communities? So we wanted to look at the different things that were colonizing them and say, are these hosts that have similar form and function being colonized by similar types of organisms? So this is our results. We find that these two different genera that are found on opposite sides of the world do seem to be colonized by organisms coming from the same phylogenetic clades, by related organisms. So here, what I'm showing you, this is an evolutionary tree of all the different bacteria that we found in our study. And in brown, along the outside, this is the relative abundance of those ones found in bog water or soil. So these were the habitats surrounding the pitcher plants. And then in blue and in red, I'm showing all of the different bacteria that were found in at least 10% of our Nepenthes or our Sarracenia samples. And I wanted them to be things that were not just randomly there, but things that were consistently found. So I decided they should be there at least 10% of the time. And what you see is that these organisms that are consistently found in pitcher plants do tend to come from the same families. So you often see the same families showing up in these pitcher plants. And we saw the same things in terms of eukaryotes, but it was more restricted. So we saw that rotifers, dipteran insects, these are the mosquitoes and the midges, and aquatic histiomatid mites also colonized regularly both the Nepenthes and the Sarracenia pitchers. So even though these are on opposite sides of the world, they seem to be colonized by similar kinds of organisms. And we were interested if that also meant that those organisms were filling similar sorts of roles within these little ecosystems. So we wanted to look at genes for prey degradation, because we thought that this would be a key sort of function within this little system. And we focused on, well, I'll be talking to you today about chitinase genes, which are the proteins that are the enzymes that are able to break down chitin, which is the main biopolymer in the insect exoskeleton. So we hypothesized that chitinase genes should be enriched in pitcher plants relative to other types of ecosystems. And I compared the metagenomes that we had for our Nepenthes and Sarracenia plants with other metagenomes that were already published for phylosphere. These are leaf surfaces, lakes, and soil habitats. And we did see that chitinases were enriched in the pitcher plant system. On the other hand, we hypothesized that cellulase enzymes should be lower in pitcher plant systems, because you don't see cellulases are breaking down cellulose, which is found in a lot of plant material, and you don't see a lot of plant material that's being broken down in the pitcher plant system. And again, we saw that in general, cellulases were lower in the pitcher plant systems than in these other environments. So it seems like these independently evolved pitchers, when you look at it really broadly and from a pulled back perspective, you can see that they seem to select for similar microbial communities with similar functions. So it's kind of an example of convergent interactions on top of the convergent evolution of the host. The second thing I want to talk about today is lab experiment that wanted to get more into the details and to tease apart these different processes that are affecting how these communities form. So the question I was asking was, when communities assemble, do historical contingencies influence composition and function? And I wanted to ask about historical contingency in a more controlled environment, because I think in general, it's more difficult to find, particularly in field sampling, because you don't know the history of that particular pitcher that's out in the wild. So you don't know exactly when it captured prey, or what the temperature gradients might be, or all the different factors that could influence how that community is formed. The common perception for bacterial communities is that environmental filtering dominates. But I think this might partly be because it's much easier to measure, to test for. And this other assumption is that composition and especially function converge under the same environmental conditions. So for example, before I go to that, I was working on this project with 2 other postdocs in Otto Cordero's lab. This was with Gabriel Leventhal and Mari Grolka. And so thinking about if composition and function converge under the same environmental conditions, there was this experiment that was done in 2018 that found that when you had microbial communities from really different soil and plant sources, they predictably converged at the family level. So across all these different types of communities, you bring them into the lab and they converge. They look very similar. But they were using single carbon sources in synthetic media. So this was not like a complex food source. There were not a lot of available niches within this experiment. And also they were using pretty stringent dilutions, so creating bottlenecks for those communities. The experiment that we did started with 10 unique pitchers that were sampled from the wild. So taking the fluid from 10 different pitchers, bringing it into the lab, filtering it through 3-micron filters to focus on the bacterial component of the community, and then growing them in little tubes and little plates in the lab. And I used sterilized acidified ground cricket and water because I wanted to use a complex medium that was a food source that was pretty similar to what these microbes might be eating in their natural habitat within the pitcher, which would be captured insects. Every 3 days, I would take half of the volume and put them into new tubes with another half volume of media. And I did this every 3 days for 21 times for 63 days. At every transfer, I would measure the carbon dioxide production with microresp. I would freeze a portion for metabarcoding to see who was there over time. Every second transfer, I would measure the chitinase activity. And this was because chitinase was what we had identified in this previous experiments as an important environmental function. And then every third transfer, we would measure a functional profile with ecoplates. So this is using a broad set of different substrates to get sort of a functional fingerprint of your community. At the end of the experiment, I cultured around 100 isolates from 5 of the 10 microcosms. And this is because I wanted to try and get individual strains separately to be able to look at their functions alone and then to connect that back to the community function. So what we found was that the communities equilibrate over time. This is showing the dissimilarity between adjacent days. So how different is the community on one day to the community at the next sampling time point. And what you see is that at first, they're very different. So from day to day, there's a lot of change happening at the beginning. And then over time, they equilibrate and there's not a lot of change happening anymore. So the communities are pretty similar from one day to the next, or from one sampling point to the next. But even though all of the 10 microcosm communities equilibrated, they still had differences in terms of the number of species that were there. So this is looking at the effective number of species, which is the exponent of Shannon diversity. And this is post day 21, so after equilibrating. And you can see that there are pretty strong differences in terms of how many different species or ASVs are there. One thing that I found that was surprising, I wasn't expecting this, but I found that the early richness predicted the final richness. So at just day 3, so after one sampling, the community had equilibrated a little bit to the lab environment, and at day 3, it already predicted the richness at the end of the experiment. And there was a very strong correlation. And this means that probably even though all of these communities are different and have different number of species, they seem to be undergoing a similar process in this lab environment because they're all in the same environment. Here we normalized by the richness at day 3 and then looked at that over time. And you can see that basically they all follow the same sort of universal rate of equilibration, which was really fascinating. These equilibrated communities are composed of different species, so they have different things that are in them. And we did not see the convergence that was found in this other experiment at the ASV level or at the family level. So you can see that there were some ASVs or some species that are present in a lot of different microcosms, whereas there are also a lot that are different among the different microcosms. I wanted to understand what are the relative effects of environmental filtering and historical contingencies. So can we see both of these acting within this experiment? Here you can see that they both do have strong effects, and I'm going to show you how I see this. So this is another NMDS plot, and day 0 is here next to the name of the microcosm. So these are all day 0, and then you see how they change over time. And you can see that there's this initial big shift, and then they sort of equilibrate. And this initial big shift is basically due to environmental selection or environmental filtering, because they're being brought from the pictures in the wild into the lab. So they're being brought into a new environment that's quite different, probably, from their picture environment. And there's this initial strong shift as they sort of get used to this new habitat. But even once they equilibrate, they remain distinct from each other. You can see that these different clustered colors are still separate. And this is due to historical contingency, because these pictures, these communities, these bacterial communities, are all being kept in exactly the same environment now. They're being given the same exact food. They're in growth chambers, they're in the same temperature, the same light regime, the same type of tubes, same size tubes, the same volume of fluid. So the only thing that's different among them is the history, is the past, is the things that have happened before they were brought into this lab environment. One thing that I found really cool was that the community assembly trajectory seems to be reproducible. And what I didn't tell you about this experiment is that I did filter a subset of the liquid, but I also kept a subset of the liquid that was not filtered, because I was curious just to see, how long would the protozoa and other small organisms last in this system. And when you look at just the bacterial component of either the filtered or the unfiltered, so the unfiltered are clear dots, you can see that they follow really similar trajectories over time. So there definitely were some differences in terms of which species or ASVs were there, but overall, they follow these really similar trajectories. And I think that's super neat, because it means that if you have enough information, this really could be predictable. One thing we were curious about was to what extent the context or the microcosm mattered, and if there were species interactions that might be affecting what we were seeing. So I'm going to walk you through this. On the y-axis here, these are all the shared ASVs, or the shared species that were found in at least 2 of the 10 microcosms. And these ones down here are all the ones that dropped out between time point 0 and time point and the first time point, basically. They dropped out from the beginning of the experiment to the first measurement point. So the dots are where that particular ASV was lost from that particular microcosm. So here, all of these shared ASVs here were not, they didn't do well in this lab experiment. And they might have already been dead or metabolically not active, because basically they dropped out immediately as soon as the experiment began. All of these ones up here seem to do really well in the lab environment. No matter what microcosm they were in, they lasted until the end of the experiment. But the majority of the shared ASVs had different dynamics depending on what microcosm they were in. So for example, this one here dropped out of microcosm 7 right at the beginning, then it dropped out of this microcosm 2 later on, but then in another one, it persisted through the end of the experiment. So most of the shared ISVs have different dynamics depending on which microcosm they're in. And this is another way to look at this. This is looking at the cosine distance between the trajectories of the same ASV, between the frequencies over time of the same ASV, and comparing that with the Bray-Curtis dissimilarity between final community compositions. So basically, this is showing that ASV trajectories are more similar when their communities that they are in are more similar. So we think that this means that the species interactions do matter, and what other organisms are there change how a particular ASV acts within that microcosm. So how does all of this relate to function? We found that there was convergence in terms of the CO2 that was being produced, and this suggests that there were really similar levels of metabolic activity across the different microcosms. So this is showing the variance in the percent carbon dioxide across the microcosms. So at the beginning, there was high variance, meaning the microcosms were really different from each other in terms of how much CO2 they were producing. But that dropped very quickly, and then it sort of stabilized. And it seemed like there was very similar levels of CO2 being produced across all of the 10 microcosms. But despite this, we found that there were still strong differences in terms of their functional fingerprints, in terms of what substrates they could use. So here, the substrate, if it's in red, it means that those microbes were really good at using that substrate, those microcosms. If it's in black, it means that they were not using that substrate. But what you can see is that each different microcosm has differences in terms of what it's able to use or not use. And the function, this functional fingerprint, strongly correlates with the composition of that community. So if the communities are more similar or more different in terms of their composition, they're also more similar or more different in terms of their function. And this is a strong correlation, both with a Procrustes correlation and a Mantel test. And I'll just say that a Procrustes analysis is basically just taking the NMDS of the composition and then the NMDS of the function, and then it rotates it until it finds the least differences between those points. So we also found that the chitinase activity differed among the microcosms. And here I'm showing you the 5 different microcosms that I ended up culturing isolates from. And you can see that particularly MO3 and MO9 had really high chitinase activity during the course of the experiment. And when we cultured different strains, we could take those cultured strains and we could measure their chitinase activity. And then we could map that back to the ASV, the corresponding ASV. And you could look at the frequency of that ASV over time in the experiment. And so what you can see is that the ASVs that corresponded to strains that had high chitinase activity were found both in MO3 and particularly in MO9. And in MO3, there was just one strain that seemed to explain the chitinase activity. But in MO9, there were 3 different strains that together explained the high chitinase activity in that community. So the functional activity of the microcosm as a whole can be explained by the activity of the individual cultured strains. To summarize these experimental results, we found that the communities change pretty rapidly in response to a new environment, the lab environment, but they still retain strong differences in their composition that was driven by historical contingencies and things that happened before we brought these pictures into the lab. The individual species dynamics seem to be dependent on the community context. So if a particular ASV persisted or was lost from a particular microcosm over the course of the experiment, seemed to depend on that community as a whole. And relevant ecosystem functions, like the chitinase activity, remain different among the microcosms. And the functional dynamics were strongly correlated with the composition dynamics and can be explained by individual strains. So coming back to this, we already had seen that environmental filtering can have a strong effect in pitcher communities. And again, in the experiment, we see that environmental filtering does play a role. But we also see a role for historical contingencies when you have a more controlled environment where you can hold steady all of these things like food source and temperature and size and shape of the tube. We also see that the different compositions of the communities do affect ecosystem function differently. So we can see that these different processes are acting within the pitcher system and that they have different relative importances. Coming back to this, when you think about the microbes that might be driving biogeochemical cycles or in large ecosystems, and then also those that might be in our own guts and affecting our own bodies, I hope you can also recognize that something as unusual as a carnivorous pitcher plant can also be used as a little model ecosystem to help us to understand a bit more about these microbial communities and how they function, how they form, and how they might affect their hosts. With that, I want to acknowledge this research was done across 3 different labs over a long time. So in Nomi Pierce's lab and Pringle's lab, Otto Cordero's lab. I had many great international collaborators in Malaysia. It was funded by different funding sources, supported by different departments, and also there were many different places that allowed me to do field work in their habitats. I also want to give a shout out to the current research that I'm doing now at Boise State. So I just started my lab here in January, and I'm going to be continuing to study the mechanisms of plant microbiome functional interactions in pitcher plants. So we just recently got an NSF microbiome theory and mechanism grant to be able to look a little bit more at these dynamics going on between the plants and their microbiomes. We're also looking at how to connect plant chemistry and effects of temperature or other environmental factors with the sagebrush leaf microbiome. So the sagebrush steppe is surrounding the area that I live in Idaho and region around here. And there's a large GEM3 grant that's happening across 3 different research institutions in Idaho. And it's focusing, one of the 2 focal study species is sagebrush. And nobody has yet really looked at the above ground microbiome. So it's a really nice system because they have very strong chemistry. They produce a lot of volatiles and a lot of toxic compounds and antimicrobial compounds. So I'm curious about how that might be structuring these communities that live on and inside of the leaves. And I'm also doing a project where I'm investigating if wheat seed endophytes impact grain quality. So this for me is a little bit of a departure because I'm used to doing sort of basic research questions, and this is much more applied, but I think it's really fun to think about how these microbes that are living inside of wheat seeds might actually affect the quality of the flour that we use to bake bread, and it could also affect our local farmers here. So with that, I want to thank all of you for coming to this seminar and for being here with me today. My email address is here if you'd like to contact me, and I'm also open to answer all of your questions. Thank you.
Thanks so much. Can we all thank Leonora virtually for a great talk? Before we get to questions, I want to remind everyone that we'll do like an informal group chat on the same Zoom link. So please stick around if you have further questions or want to talk more. Otherwise, feel free to unmute yourself for a question, or we'll keep an eye on the chat as well.
There's one question in the chat. Do you want me to read it?
Sure.
Dean Walton asks, this is more of a field question. Do different food sources, insect species, bring in or drive different communities to any degree too?
Yeah, that's a good question. And it hasn't really been investigated in detail. So there was an experiment that I did early on in my PhD where I was using sort of sterilized gauze to cover the pitchers and to see if you still had a lot of colonization by microbes even without any insect prey or insect associates. And I did still see a lot of microbes colonizing those pitchers, but I do think that there probably are differences in terms of what's brought in by prey and then also the compounds that are in the prey. So the main prey across different types of pitcher plants is usually ants. So for both the Sericinia and the Nepenthes, usually ants are the main insect prey. But you do find lots of other things that are in there too, like grasshoppers or beetles or all kinds of other things. So I think that when you get something like a big fatty beetle, it's going to have probably a really different effect than when you have something like just a small ant. And you do definitely see putrefaction that can happen in the pitchers. So if something that's sort of too big falls in, it can basically shift that ecosystem and make it anoxic, and you get kind of overgrowth by just a particular type of bacteria. Sometimes that can actually sort of kill the leaf, but it does seem like it sort of shifts it to a not as healthy microbiome. And there's one species of Nepenthes that are found in Southeast Asia that associate with ants as well. So it's an ant plant in addition to being like a carnivorous plant. And those ants that live, they live in this swollen tendril, these hollow tendrils of the Nepenthes species. And they can actually like crawl down into the liquid, they can swim around and they can climb back out. So they're not, they seem to be able to avoid a lot of like being trapped and digested. And sometimes what they'll do is if something really big falls in, like a cockroach, they will like dive in and dig it, like pull it out, and they pull it up to the top of the pitcher, and they'll actually like break it apart and like eat parts of it. And then it seems like it's easier for the plant to digest. So there's sort of these funny different interactions that can happen. I think it would be interesting to look more in exactly how different prey species shift the communities, but it hasn't really been done yet.
Do you see the other questions in the chat, Karen?
I think if I move out of, if I stop sharing my screen, then.
Yeah, here's one from Caitlin. Is there seasonal variation in the pH levels within the same species of pitcher plants? Was there significant taxonomic variability in the microbiomes between pitcher plants with the same species?
Okay, this last one from Caitlin Smith.
Yeah.
So in terms of seasonal variation, not necessarily, but there definitely is sort of developmental variation. So what you see is that only certain species of Nepenthes will get really acidic and other species won't. And the ones that do get really acidic, they generally start out at sort of a medium level, and then they get very acidic, and then they slowly come back up. And it does seem to be kind of predictable. So the plant does seem to do that over time from pitcher opening to sort of older pitchers. I found that just in a growth chamber study that I looked at, just sort of preliminary like pilot data. But then I also found an older study from a lab in China that had found the same thing. And was there significant taxonomic variability in the microbiomes between pitcher plants within the same species? Yeah, there is a lot of variability in general in the pitcher plants. So from one pitcher to another, there's a ton of variability. And I think there are just so many different things that can be driving that, right? So there can be succession, and you don't always know when a pitcher's open. So when I was sampling, I was trying to sample across pitchers at different ages because I knew that succession, you know, after we did that other experiment, we knew that succession was affecting the communities. And then also there's so many other things like when prey was captured, you know, which microbes arrived there. So you do see a lot of variability even within one species across different pitchers.
There's also a question or a couple questions from Monica about whether Nepenthes and Sarracenia evolved in different biogeographic regions.
Yeah, so the Nepenthes evolved in Southeast Asia, and the Sarracenia evolved in the Americas, that family. So in the Sarracenia, there's one genus that's found in South America, so Venezuela, like on the Tepuis of Venezuela, these are Heliamphora. Then there's Darlingtonia, which is found like in California and Oregon. And then the Sarracenia are found along the Gulf Coast and then up into Canada. So that's in the US. And in Southeast Asia, it's not totally clear where that group evolved, but you find them from Madagascar, some in India. Most of them are in sort of Southeast Asia and Malaysia, Singapore, Thailand, the Philippines, and then they go all the way to Australia. Okay, and then for less acidic pitcher plants, how do they digest their prey? Is it that they rely solely on chitinase from bacterial communities? How is the macroorganisms such as mosquito larvae withstand the chitinase in the pitcher? So those are a lot of really good questions. Thank you for all those questions. It's thought that, at least in the Nepenthes, there was a study that showed that the proteases in the Nepenthes work better at these really low acidity, like this very acidic pitchers. So I think a pH of like 2 or 3 was optimal for some of those proteases found that the plant was producing. And I think that there might be a trade-off. This is a paper that I have not published yet, but it was a project I did with an undergrad. And what we found was that the less acidic pitchers had a lot of nitrogen fixers. And we don't know for sure that they were active, but we found a lot of the NIF genes in the less acidic pitchers and then not in the very acidic ones. And I think there could be a trade-off in the sense that these microbes might actually potentially be fixing nitrogen within the pitcher. And so maybe they're actually helping out more, the bacteria are helping out more in the less acidic pitchers, and in the more acidic pitchers, the plant is sort of doing it itself more. So that's a theory I have. And in the Nepenthes, they make their own chitinase too. So they can make their own chitinases. There's this idea that maybe they make more of their own enzymes when they first open, and then once they have a community established, they make less of their own enzymes. But that hasn't really been shown for sure. The Sarracenia do not make chitinases, but they do make some proteases. And how do macroorganisms such as the mosquito larvae withstand the chitinase in the pitcher? So I think a lot of these enzymes are basically only acting on things that are dead, right? So, so you only really see the degradation happening when things die and don't have their own sort of bodily functions that are maintaining that integrity of the organism. And I think that some of the insect larvae that are in those pitchers, like the midges, they kind of pull apart and will sort of tear apart the things that fall in. And I think then those enzymes have easier access to the compounds inside of the insects. But it still is not clear. I do think there probably are some special adaptations that some of these pitcher plant mosquitoes have for being able to survive in these environments. Because one sort of other natural experiment that we've seen is, for example, Sarracenia purpurea, which is found in the US, it was introduced to Switzerland 100 years ago. But in that 100 years that they've been thriving in Europe, there are no insects that have colonized and started living in the pitchers. So you don't see any local mosquitoes or midges that have transitioned to surviving in these pitchers there. And I think that that could signify that, maybe there is a bit of adaptation that is needed to be able to really colonize and live in these pitchers. Oh, Raven asked, is the little guy going to be eaten? You're thinking about the frog, right? The frogs don't get eaten, at least usually they don't. I've seen frogs hanging out in pitchers a few times, and I think they use them as protection during the day from sunlight, and it's sort of like a little hiding spot. But they're pretty good at getting out again. They just sit on the edge, on the lip of the pitcher, and they don't go all the way down. Although in some pitchers, there was a paper that came out recently, and I have seen this myself. Sometimes salamanders do get eaten by the Sarracenia purpurea. And it does usually putrefy the pitcher, so it gets really stinky. I don't think that they're really, I don't think it's really meant to be their optimal prey source, but you do see salamanders occasionally getting trapped in the pitchers. In the Nepenthes in Southeast Asia, there's actually some frogs that have specialized and only lay their eggs in pitcher environments. So there are some pitcher plant frogs that live in the Southeast Asian ones.
Hi, Leonora, I have a question, if I may. I think there's no other questions in the chat, so I'm grabbing the opportunity. Thanks for the great talk. I really enjoyed it. Very interesting stuff. I actually worked on bromeliads a long time ago, so I definitely appreciate using these plants as model systems.
I know.
Great. I had a question about a very interesting figure that you showed. Unfortunately, you didn't spend too much time on it. You showed that there was a strong correlation between the final community composition, and correct me if I'm wrong in my interpretation, you showed a strong correlation between the final community composition and the individual trajectories of ASVs. And then you sort of pointed out that that implies community interactions. And again, correct me if I'm wrong, but the way I see it is that in principle, it could be possible that the trajectories of ASVs are actually independent of one another, so that there's actually zero interactions, and that could still lead to the pattern that you saw, since it's a bit circular. If trajectories vary randomly from pitcher plant to pitcher plant, then you're still going to see potentially a strong correlation between the final community composition and the individual trajectories, since the community composition at the end is influenced by the trajectories. So have you done some sort of a null model analysis testing whether that strong correlation is indeed not explained by such a arguably simplistic null model, but in principle it is possible?
Yeah, we did a null model test, and it was significant compared to the null model. It's in the Nature Communications paper, and Marigosa led that analysis.
Cool, very cool. Thanks.
Are there any other questions? If not, can we thank Leonora again? And I'm going to suggest that we come back at, say, 1:20 for our group chat and take a break to make some tea or whatever you need. And we'll be on the same Zoom link.
Thanks, everyone.